So I'm going to talk about marker-assisted selection. There's 3 generations of marker-assisted selection: the marker-assisted backcrossing, the marker-assisted recurrent selection, and the genomic selection. The first one, the first generation marker-assisted selection, so you have the same scheme. This scheme that I presented earlier will be repeated for each of those generation of marker-assisted selection. This first generation, from the species diversity and the locally adapted material, you'll develop an experimental depopulation on which you will study the relationship between the phenotype and the genotype. Basically, some association mapping or some mapping, QTL mapping. And from this association, you will define markers that are significantly associated with genes or QTL of interest. And then the idea is to process on marker-assisted selection on the material that is under selection. So not completely the same material that is used for the genetic mapping. This concept is based on the linkage equilibrium between marker and genes or QTL, which is, I mean, this linkage equilibrium, which is involved in the variation of simple or quantitative traits. I don't know. I don't know if I wish I could see you. If you are aware of the concept of linkage equilibrium, which refer to the non-random statistical association between alleles at 2 loci, which basically mean that the loci A and the loci B will always segregate together in a progeny. So it's defined, the linkage equilibrium is defined by 2 indicators, a D and R, which are basically 2 different formula which are pretty much correlated. So if you have no linkage disequilibrium, which means you have a linkage equilibrium, that means the alleles at the 2 loci will be segregating independently. So you have no linkage. If you have a tight linkage disequilibrium, it means that always the small a and small b will be segregating together. That means that there's a link between those 2 loci. It is important to know the linkage equilibrium estimated in the genome, because that will tell you how many numbers you need to really characterize the genomic diversity of your species. Basically, if you have very tight, big linkage equilibrium, that means that you have blocks of chromosome that are segregating together. So if you have these blocks, that big blocks, that means that you don't require many markers to really characterize your genomes. If you have very tiny linkage equilibrium, for example, 400 base pair, that means that you will require many molecular markers to really see the diversity at the genome level, to really pinpoint the diversity of the recombination. So the first generation of markers, so that was a small apartheid to really make sure that the concept of linkage equilibrium is understood, because this is what makes the first generation of marker-assisted selection possible. And this is an example with the SUB1 gene. SUB1 is a gene or QTL, that was discovered in 1996 in a population segregating for the ability to sustain submergence. It was mapped in a population of I don't have the parental lines here. Well, they are the 2 arrow on a histogram. The QTL mapping reveal a QTL that explain 90% of the phenotypic variance. So the progeny with the QTL sustain submergence, and they had this marker, which is associated to the QTL, This is a molecular marker here. And the one that do not have the molecular marker were susceptible to submergence. So basically, the idea was to integrate this sub-1 QTL into a susceptible line, which improves significantly the tolerance to submergence. So it took a while from 96 to 2013, about 17 years, to really make a difference in submergence tolerance owing to the integration of sub-1. So the backcrossing is when you have a recurrent parent like Schwerner is susceptible, so it's a very good material but susceptible to submergence, and you have a donor that is not necessarily good looking but has a high tolerance to submergence. So you develop the F1, the hybrid plant, and then you backcross to the recurrent parent in order to have the visual uh, statute close to Schwerner, which is a germplasm that you want to have in the field, but with the trait of interest of the donor parent. So you have this process of backcrossing that you repeat many, many times, and at the end you have a genome that really resemble to the recurrent parent, but has the gene of interest coming from the donor. The problem is that you always have remaining things from the donor that you may not want, and this is due to the linkage disequilibrium, because you may have some genes that will be linked to the sub-1 QTL, and they will be dragged along with the sub-1. So what has been done in order to avoid that is marker-assisted backcrossing, where basically you process the same way, but early on you make a marker-assisted selection to select the plant that has a favorable allele at sub-1, which here is a marker associated to sub-1, but has the lowest genetic background from the donor, so that you don't want all the undesirable traits from the donor. And then you have different steps of backcrossing in order to remove all what is in the background genome that belongs to the donor, but that you don't want because you want to really recover the recurrent parent. So you have different steps of backcrossing, and you have with a very high percentage of recurrent parent in a very early generation of backcrossing. So basically, in a small graph on the bottom right, you see the conventional backcrossing without marker-assisted breeding, and then which is a lower line, and the upper line with the square is a marker-assisted backcrossing, which allow you to recover on the y-axis the percentage of recurrent parent genome that is recovered along the different backcrossing, which is the x-axis. So that was the first generation of marker-assisted selection. Now we are going to move on to the second generation, which is basically different because here you build the relation genotype-phenotype directly for the unit under selection. So you create a new variability using the species diversity and the locally adapted material, and you establish the relationship phenotype-genotype in this material, which is a material under selection. And with this relationship, you are very close to the material, the candidate unit for selection. And here you make selection, marker-assisted selection. Oh, sorry, this is a French slide, for the QTL under selection. Here is an example. So you have elite donors, you make crosses, and then you have a population that segregate with the two genomes, the parental genomes. So here you do QTL mapping, and you can detect different loci associated to different traits of interest. And so in the progenies, what you can do is to select directly the progeny that you want to cross again, but by selecting with the presence of the key marker that are associated with key trait of interest. So basically, you define a trait value based on the marker, presence, absence of the marker of the alleles, favorable alleles in the progenies. So you do an accumulation of favorable alleles for the QTL of interest, and then you recombine those entries. And then you do that several times, marker associated selection of best recombination, and then recombination. So basically, this repetition will be driving your breeding program to apply the marker assisted recurrent selection, where you will build the ideotype of interest by selecting the progenies which have the favorable alleles at all the key QTL that you want to include in your new varieties. So the expected result is to save time and to build new IDEO genotypes. So it's really a change in focus where you identify the combination of the favorable segment of the genome, and you accumulate this segment in the new varieties. I may skip this one. Uh, the marker-assisted recurrent selection, so we have few examples. For example, for drought tolerance. So this is an experiment where there was a cross between two parents and then development of progenies. Those progenies were evaluated into different conditions because the objective was to target drought tolerance, but also yield potential under different topo sequences of hydrological conditions. And so the phenotyping and the genotyping of these different lines developed from these crosses allow to do QTL mapping, and thus the distinction of the favorable alleles that have to be combined to develop the optimal cultivars. And basically, uh, selection of cultivars, recombination of the best one, allow to develop new elite lines combining the key alleles at the key trait of interest. So that was the marker assisted recurrent selection. Now I'm going to talk about the third generation marker assisted selection, which is the genomic selection. And this is a completely different story here. We have the species diversity, the locally adapted material, and this will develop or constitute a calibration population. This calibration population will be phenotype and densely genotype. And then on this population will develop the genomic estimating breeding value with all the markers that have been used for characterization the characterization of this population. And thus basically what remains to do is to just genotype the selection candidate units and to evaluate their breeding value using the same genotyping. So this is a very big change where from marker-assisted selection to genomic selection, we went from identifying significant marker to estimate their effect on the target trait and select on the sole significant markers to build ideotype with the limited number of markers, to genomic selection where you estimate the effect of all the markers, so potentially several thousands of markers, and predict the performance using all the genetic variants to select the genotype based on the value of all the markers. So this is a very big change in the concept that we use the entire genetic information to develop new, to select new elite lines. So the genomic selection has a very high potential in terms of the delta G, which is the genetic gain. This is a breeder equation with delta G, the genetic gain, as a function of the intensity of selection, selection accuracy, and genetic variances over the generation interval. The principle of genomic selection, so as I said, you have a population, a reference population that is genotype and phenotype. You develop prediction model, statistical model that allows to develop to estimate a random marker effect. So for each marker, you will have an effect. And then this marker effect will be used on a new population, which is a candidate population, which will be genotyped with the same set of marker. And with the information of the marker effect, we will calculate the genomic estimated breeding value of each individual. Basically, those individuals will never be phenotype, they will just be genotype. And owing to the calibration model developed with the reference population, we will be able to estimate their breeding value and select the best entries for selection. So this is a very new method that has been developed in early 2000 and is still currently in use. So I'm not going to explain, probably in a different lecture, the way we do the job in order to calibrate the model and estimate the relevance of the model. We have done, I may just go directly on the example. This is also the estimation of the precision of the prediction using a candidate population to validate the prediction model. So once we have estimated the genomic estimated breeding value, we may still phenotype to validate the accuracy of prediction by calculating the correlation between the GEBV and the true phenotypic information, so that we'll make a robust assess the robustness of our model.